Photosynthesis lecture from chapter 7. In this lecture, we're going to look at specifically the light reactions. We'll learn that photosynthesis can be broken down into two reactions, the light reactions and the Calvin cycle, otherwise known as the dark reactions. We'll look at that in part 2 of the photosynthesis lectures. Let's begin with arguably one of the most important reactions on the planet, and that is the photosynthetic equation. So notice that there are 6 CO2 molecules, 6 water molecules, and then the role of sunlight, which is to provide the energy to create glucose and oxygen. There's quite a bit going on in this reaction. It doesn't happen in one single step. In fact, it doesn't really happen in the 2 steps, light reactions and Calvin cycle. And I say that because the light reactions is a series of multiple steps, just like the Calvin cycle is also a series of multiple steps. So this reaction that we're looking at here on the leaf is a summary of all of those reactions. Our task in chapter 7 is to appreciate all those little steps, but we don't want to just memorize the steps or just appreciate them for their complexities. We want to appreciate them in the context of chapter 5 when we covered energy. We want, in other words, an opportunity to explore the concepts of energy of chapter 5 in the context of photosynthesis. In fact, we are also going to look at diffusion and osmosis, the laws of thermodynamics, and enzymes. All of these concepts of chapter 5 are important to understand before moving on to this chapter on photosynthesis. But I do want you to know the photosynthetic equation. It will be on your second unit exam. So let's just make sure that we know the reactants. That would be CO2 and water, and that we know the products. Those would be glucose and, again, oxygen. Okay, there's going to be a lot more to say about this, right? So let's get to it. Let's look at the photosynthetic part of any plant, and that would be in the leaves by and large. The reason why leaves are so important is because that's where photosynthesis is taking place. Notice in this image here that the leaves are, like most leaves, flat, thin, and that increases the surface area, right? Which is great for trapping a greater amount of light, yeah? To take a leaf and flatten it out and expose it to the sunlight increases the amount of light they can absorb. But there's a drawback to that strategy, and that is, of course, that the leaf can also dehydrate. And as you saw in the previous slide, a leaf depends on not just CO2 for creating sugars, but also water. So it needs that water not just for photosynthesis, but also to keep itself cool as it's exposed all day to a sun. All right. If we were to take a leaf and produce some sort of cross-section so that we can look into the leaf at the various layers that make it up, what we would find is that at the top layer here, we have what I like to call the upper epidermis. Okay, I'm working on my penmanship because that was a big deal in the previous presentations I started with. I want you to know that the upper epidermis is important because it produces a wax. In fact, we call it a waxy cuticle. All right, I like my penmanship already. All right, so what's up with this waxy cuticle? I think if I ever were to give you a leaf and ask you which is the top of the leaf and the bottom of the leaf, I think most of you would be able to tell me which is the top and which is the bottom. And I think most of you would look for some sort of glossy finish to the leaf, right? That glossy finish is basically the waxy cuticle. So why does the top of the leaf produce this waxy coating? Why is there less of it on the bottom? The bottom is what I call the lower epidermis. Well, the, uh, the top of the leaf is important to add a wax layer to because of things like wind, right? It turns out that on a really windy day, a leaf can get dehydrated much faster than on a really, really hot, dry day. And I think you also have experience with this, right? In what season are you more likely to put on skin lotion and chapstick? I would argue that that's probably in the fall when we tend to have, dry, windy conditions, and not so much during the summer, right? So this is the strategy employed by the leaf. The leaf is trying to prevent dehydration or the loss of moisture from its leaves, which are thin and flat. Remember the conversation on high surface area to low volume ratios. So the leaf is essentially designed to trap a lot of sunlight, but it's also unfortunately going to lose a lot of moisture as well. And the wax is there to prevent the loss of moisture, not only on hot, dry days, but more so in the fall when the wind tends to be dry and there's a lot of it. Okay, so we know the upper epidermis is waxy to prevent dehydration. Okay, what about the next layer below that? This layer of cells right here. That layer is called the palisade mesophyll. And you see the word here also. Mesophyll stands for middle of the leaf. That's literally what it says. Meso, middle, and then phyll is a leaf. The palisade mesophyll is important because it is jam-packed with cells. You can see them here. They're kind of oval, and they are really concentrated. I mean, they are really, really jammed into each other. And notice that every cell also has a lot of chloroplast. And because it's the one that gets the first incoming input of light, this layer is important to the leaf because it does most of the photosynthesis. So I want you to know that about the palisade mesophyll. Most of the photosynthesis is occurring in this layer. All right, what about the next layer below? This one here. Notice that in this layer, the cells are more loosely aggregated or collected. In other words, they're not jam-packed like the palisade mesophyll. There's a reason for that. In fact, we describe this layer as the spongy mesophyll. Again, mesophyll meaning middle of the leaf. So this word will be easy, I think, to remember. And spongy is, again, to remind us that there's a lot of air spaces, right, between all of the cells of the spongy mesophyll. So why is it spongy? Well, it's spongy because of these two important gases, CO2 and oxygen. Notice that really there's one major entrance into the leaf, and that's through these openings produced by two cells that form what we call a stoma. I'm going to draw those two cells over here and show you what they would look like when they are opened. They look kind of like kidney bean shaped cells, right? And CO2 would go in and oxygen would come out. Well, do you remember where I said that the palisade, I'm sorry, I just gave away the answer. I was going to ask, do you remember where I said that most of the photosynthesis is occurring? It's occurring in this layer, right? The palisade mesophyll. So we need to get CO2 to this region of the leaf in high amounts very quickly as we can. And also because this is where most photosynthesis is occurring, there's a lot of oxygen that's being produced. Again, we learned about these two gases in the previous slide when we were looking at the photosynthetic equation. So my point is this: if you need CO2 to be delivered really quickly to this group of cells, how do you accomplish that if you have to travel through each cell to get to the palisade mesophyll? In other words, imagine if the spongy mesophyll was just jam-packed with cells. That would mean that CO2 would go in, and then immediately, after traveling through air, which we learned in diffusion and osmosis to be very, very fast for diffusion, after traveling through air and it gets in through the stoma, it's now inside a cell. And that cell has a liquid in it that is the consistency of honey. That's going to slow down CO2 quite a bit. Now imagine the CO2 finally gets out of that cell, and then it gets into another cell. And now that cell is going to slow it down, and then it goes into another cell and another cell before eventually getting to where it's needed. Do you see that that's going to be super slow? Diffusing through a liquid medium that is a consistency of honey is intensely slow. And doesn't that defeat the purpose of being flat as a leaf to maximize photosynthesis? It does. So the spongy mesophyll is spongy for no better reason than to have air spaces that allow the CO2 to get where it needs to be as efficiently as possible. In other words, it's much easier to travel through air than any other medium. And that's why the spongy mesophyll is spongy. The same holds true for oxygen, right? Oxygen is being produced, again, by the palisade mesophyll. So that oxygen needs to get out of the leaf quickly. What if it were taking forever to get out of the leaf and photosynthesis was occurring at a fast rate anyway? Well, it would just be a matter of time before the internal pressure, the air pressure of oxygen in the leaf, would begin to damage the leaf, right? So the spongy mesophyll is spongy to allow the rapid diffusion of gases. So I want you to know that about the spongy mesophyll. And we have now applied something that we learned in Chapter 5, diffusion, in the context of photosynthesis, or at least the structure of an anatomy of the leaf. Okay, so the stomate, remember, the stomate would be plural, stoma, right? That's one opening produced by two cells that have the shape of kidney beans. And those two cells are called guard cells. And that's a cool name because they sort of do guard the entrance into the leaf, right? That's basically how a leaf breathes, yeah? Except that it does the reverse breathing that we do. It breathes in CO2 and it breathes out or exhales out oxygen. Okay, so let's now focus on the very important chloroplast, right? This one right here. Notice that the chloroplast has several membranes. It's got three, okay? It's got an inner and outer membrane. And you might recall from our earlier discussion in Unit 1, Chapter 4, the endosymbiotic theory, the theory that the chloroplast was once a free-living bacteria. We think that that's what explains this outer membrane. But the membrane that we really want to focus on for photosynthesis is this one here, the thylakoid membrane. And you're going to know that membrane really, really well. It's all over the place. So I'm not worried about that one. Notice that the thylakoid membrane here seems to be just very, very deeply folded membrane, right? At first glance, it appears like discs stacked on each other, yeah? But in reality, it's just one single membrane, really folded tightly, efficiently in a very tight compartment. And again, that's to increase surface area to volume ratios, and that in turn increases the efficiency of photosynthesis by one single chloroplast. And look at how many chloroplasts a cell would have, right? That's quite a bit. So the whole strategy, the whole idea, the whole design of a leaf is to really, really drive photosynthesis as fast as possible. All right, so I want you to know about the thylakoid membrane. I'll say more about this in a different slide. The other portion of the chloroplast I want you to know really well is the stroma. The stroma is the liquid inside of the chloroplast. It's sort of similar to the liquid in the cell. The liquid in the cell is called the cytosol, if you remember. The liquid in the chloroplast that bathes all of this thylakoid membrane, all of that green membrane is completely surrounded by a liquid called the stroma. So I want you to know both of these compartments, the thylakoid and the stroma, because we're going to break up our photosynthesis into two steps. The first step will take place in the thylakoid membrane. The second step will take place in the stroma. One final comment. Notice that the membrane that appears to be in stacks, like here, is called a grana, right? The stack is a grana, but that's just the name for the stack. It's still thylakoid, right? All of it is still thylakoid membrane everywhere. You can see it's all attached to each other, but we call a stack a granum, and that might come up in a later conversation, maybe in your photosynthesis lab. Okay, let's move on now. I want to outline the two steps of photosynthesis. I'm going to give you some advice, and the advice is that you get yourself a flashcard, two of them in fact, and you give one the title of the light reactions on one side of that card. And on the opposite side of that card, you're going to then put in an outline that shows what is required for the light reactions and what is produced by the light reactions. In your second index card, you're going to put on one side the Calvin cycle, and on the opposite side, you're going to create again an outline that shows what is required for the Calvin cycle and what is produced by the Calvin cycle. So I'm going to do that here on this blank slide. So one, the light reactions. All right, so this is what you would put on the front of the card. And in the back, I want you to write what are the reactants? In other words, what goes in. And on the other side, you're going to write the products. In other words, what comes out of the light reactions. All right, so what are the reactants? Well, if we go back, we know that we're going to need light. So what is the role of light again? It's just to provide energy, right? In the form of sunlight. That's one thing we need. What's the second thing we need? We need water. Well, what does water contribute to the light reactions? We're going to learn that water contributes electrons. This is very important. In fact, if you should water your plants at home, I want you to practice applying what you're learning here. You're not just hydrating your plants. Water has a deeper value to a plant, and that is the electrons that are in water. So you're actually not just hydrating your plant, you are providing your plant with a source of electrons so that it can carry out photosynthesis and feed itself. Okay, what else would we need? We're going to need a third item. We're going to need a molecule called NADP. It's got a plus on it. And there's a fourth thing we're going to need. We're going to need adenosine diphosphate. You remember that to be part of the ATP cycle. And you're also going to need a phosphate that is a functional group. It's not attached to any organic molecule. And for that reason, we call it an inorganic phosphate. So here I'm just going to draw it like the way I would normally draw it in lecture, and I have drawn in lecture. It's just a phosphate inside of a circle. Okay, so we need these four items. Okay, so once photosynthesis takes place in the light reactions, and we should remember that the light reactions happen in the, do you remember? That's right, it's in the thylakoid membrane, that really folded up green membrane, right? You want to know this. One of the questions on your second test will be, where do the light reactions take place? That's a super easy answer, right? We should all get that. Other questions you will be asked are what are the reactants, or which of the following are not reactants of the light reactions, or which of the following are products of the light reactions, or which of the following are not products of the light reactions, right? Okay, so what happens when you provide light and water to a plant? That water is going to become 1/2 of oxygen plus some acid ions. You remember H plus is acid, right? So the light reactions produce acid, and that acid is very important, as you will see later. This oxygen is one single oxygen, right? 1/2 of 2 is 1, and that's the one oxygen there. But in chemistry, oxygen doesn't exist by itself. It exists normally attached to another, O2, but we need to be specific, and so that's why we say, okay, it's actually 1/2 of O2, one single oxygen, this one right here in water. Okay, what happens to NADP plus? Well, it becomes NADP with an H. All right, here is a really good practice moment for you. Do you remember that in chapter 5 we discussed where a chemical stores its energy? And we agreed that all chemicals store their energy in bonds. So my question to you, my practice question to you would be, which of these two, NADP plus or NADPH, has a greater amount of energy? Your answer should be NADPH, and that's because it has a hydrogen that NADP plus does not have. In order to have that hydrogen, you need to be stuck to it. That means you need a bond. So NADPH has one bond that NADP plus does not have. And for that reason, NADPH has more energy than NADP plus. Great. Let's practice again with ADP and phosphate. What happens to ADP and phosphate? It becomes ATP, that energy molecule of the cell. All right, so once again, who has more energy? ATP or ADP, say by itself, or even with the phosphate. We would argue that ATP has more energy. Why? Because it has an extra bond that ADP does not have. Okay, so this was a good practice moment. All right, so you might be wondering, okay, well, what happens with this energy? What becomes of it? Well, that energy was used to split water and to excite electrons, and we will see that in subsequent slides. Okay, so in summary, I think that if you create an index card that has the light reactions and in the thylakoid membrane on one side, and then reactants and products, this whole outline on the back side, you will have an effective card that will help you answer several questions on the test. Okay, let's look at the second set of reactions. These are called the Calvin cycle. They're also known as the dark reactions. But I kind of prefer the Calvin cycle over the dark reactions because, in all honesty, the dark reactions cannot happen unless light supplied them with some materials. So I prefer we just call it the Calvin cycle. So where does the Calvin cycle take place? This one takes place in the stroma. Do you remember what the stroma is? The stroma is the liquid inside the chloroplast. Students will confuse the stroma with a stoma, and that makes total sense to me. Do you remember what the stoma is? The stoma is the opening of the leaves, right, made up of 2 guard cells. All right, so you can go back to the previous slide and you can sort that out. All right, so again, what are the reactants? In other words, what goes in? And what are the products? In other words, what comes out? Um, well, the reactants of the Calvin cycle include CO2. Okay, we're going to need 3 of those guys. And we're also going to need NADPH. And guess where that comes from? That's right. It comes from the light reactions. So do you see that the light reactions are now feeding the Calvin cycle, right? We're also going to need ATP. And guess where that comes from? That's right, also from from the light reactions. And once the Calvin cycle gets these 3 reactants, these 3 ingredients, what happens to them? Well, the 3 CO2 molecules are going to be turned into a molecule called glyceraldehyde 3-phosphate. Otherwise, it's just a 3-carbon sugar. Okay, so in all honesty, photosynthesis really produces a 3-carbon sugar. It's not really glucose. In fact, most plants will produce the fructose before they produce the glucose. And then, eventually produce glucose, attach it to fructose, and create what we call sap, or, the sort of sugar, granular sugar that you use at home. Okay, we'll talk more about this Calvin cycle in the second lecture for photosynthesis, right? But let's finish up this sort of flashcard moment we're creating. Anybody want to take a guess as to what happens to NADPH in the Calvin cycle? If you said it goes back to being NADP with a plus, you are correct. NADPH is broken down to create NADP plus. And again, it's now with less energy than when it was in the form called NADPH. And finally, ATP, I think you can guess, right? What's going to happen? It'll become ADP and a loose phosphate. And that means ATP will be broken down, so it will lose its energy when we break one of its bonds, and it becomes ADP with a phosphate. Okay, again, we'll talk more details about these 2 reactions and in 2 lectures, the light reactions and the Calvin cycle. Okay, in this slide, I want to add to your growing understanding of important reactions in a cell. In chapter 3, we learned about dehydration synthesis and hydrolysis. So in summary, dehydration synthesis is the reaction used by cells to create larger polymers or larger molecules. And hydrolysis is used by cells to break down large polymers into individual monomers. That was chapter 3. In chapter 5, we learned about exergonic and endergonic reactions. And I think we should maybe practice some of that here as well. See here, there's 6 CO2s. Those 6 CO2s are loose, right? There's 6 CO2 molecules wandering about. And if you follow the blue arrow, you'll see that the 6 CO2s are combined to form sugar, C6H12O6. So, is this reaction a reaction that requires energy, or is this a reaction that frees up energy, gives away energy? In other words, my question is this reaction endergonic, needing energy, or exergonic, giving away energy? The answer is it's endergonic because you took 6 loose things and you attached them to each other. You created bonds. And now the energy is stored in the bonds. So you needed energy to store it in those bonds. You needed energy to assemble the 6 CO2s to create something bigger. Dehydration synthesis is an example of an endergonic reaction, a reaction that requires energy. All right, that's basically photosynthesis. Photosynthesis is a largely endergonic, energy requiring process. This is why the sunlight is so important, because photosynthesis is something all living things rely on. And fortunately for us, the sun is providing a wealth of energy to be used to create all that sugar that feeds the rest of the world. Let's look at the reaction on the bottom. This is sugar here, and notice that if we follow the red arrow, it's now all CO2, loose. They're not together anymore. That means that you broke a bunch of bonds. You did the hydrolysis reaction now, and that's a breakdown reaction. And when you break down molecules, you break their bonds, and those bonds release their energy. So this reaction, right, is exergonic. It releases energy. And you knew that in your own way. You know that if you consume sugars, you are consuming an energy molecule, and that when you break it down, it gives you its energy, right? So breakdown, hydrolysis, exergonic, right? Because you broke bonds. All right, so that's a summary of chapters 3, dehydration synthesis and hydrolysis, and chapter 5's endergonic, exergonic reactions. But now we're in chapter 7, and we're going to add another reaction that I want you to know. And that reaction is called oxidation and reduction. So you've heard of oxidation, I'm sure. You heard of iron oxidizing. Metals oxidize all the time. What does that mean? Oxidation is the loss of electrons. But oftentimes in a reaction like this one here, you don't see the loss of electrons. In fact, there's no mention of an electron anywhere in this reaction. So we need to have something else to help us understand what happened to electrons, who lost them, who gained them, that sort of thing. So the loss of electrons can also be thought of as a loss of hydrogens. Okay. All right, so that's oxidation. Pretty simple. Anytime you see a molecule losing electrons or losing hydrogens more often, then you can say that molecule has been oxidized. So when iron was oxidizing or rusting, it was losing its electrons. It was losing hydrogens. What about reduction? What is that? Well, I think you can probably guess that it's going to be the opposite of oxidation. Reduction is the gain or the gaining of electrons. And again, because we don't always see the electrons in a reaction, we have to look for something else to help us understand what's happening, and that's going to be the gaining of hydrogens. All right. Okay, so here's the final comment on this, and then we'll start applying it to these two equations here, this one and this one, all right? We're going to apply what we learned. It turns out that molecules don't just misplace their electrons. They don't just like all of a sudden lose them, like, oh, where'd it go? I forgot where I left it. And molecules don't just gain electrons because they found them laying around. It turns out that a molecule loses an electron because something else took it from it. So do you remember us discussing in chapter 2 how in an ionic bond, one atom pulled away an electron from another atom, and that this is how we created ions, and then this is why opposite ions attract each other? That was oxidation and reduction. That was the loss of an electron and the gain of an electron, right? And that was because of something that we call electronegativity. I'll write it here to remind you. And you can go back and read this part if you need to. Electronegativity. Remember that this was our way to understand that some atoms are better at holding electrons than others. So it turns out that oxidation and reduction happen simultaneously. As something loses an electron, something else is gaining it. In fact, one is stripping it from the other, just like what we saw in an ionic bond. And because they happen simultaneously, we call that process redox, as in reduction and oxidation happening at the same exact time. Okay? All right, so I want to talk, before I move on to applying what we've learned, I want to talk about one potential pitfall you might run into, and that is in the word reduction. The word reduction to most students implies losing something. You've been reduced as in something was lost. But it's not, it's gaining, right? So a mnemonic that our chemistry students use, we all used when we took chemistry and we learned about redox, is a mnemonic called oil rig. I'll write it here. Oil rig. And it basically says this: oxidation is loss, reduction is gain. Oil rig. And that might help you on a test. In fact, I've seen my students write it on the test, oil rig, just so that they can jog their memories when they get to a question about oxidation and reduction. Okay, I think we should apply it now. Let's go to this equation right here, right? Look at CO2, and then let's follow this blue arrow. And what happens to CO2? CO2 all of a sudden appears with, it still has carbon, like here. It still has oxygen, like here. But all of a sudden, there's these hydrogens there that weren't present before. So what we should understand here is that CO2 has gained hydrogens. So what happened to CO2? If it gained hydrogens, CO2 must have been reduced. That means it gained electrons also, right? Remember, you cannot have reduction without oxidation. So who provided the electrons? Do you remember in the previous slide we said that water was not just to hydrate your plant, but to provide it with a source of electrons? Well, here it is. Here's now water, H2O, and let's follow its path. It becomes oxygen. No hydrogens. That means it lost hydrogens. That means it lost electrons. All right, I'm pretty happy with myself with that explanation. I feel like everybody should understand that pretty well. Here's one question that a lot of students will ask. If this reaction right here is photosynthesis, what's up with this water? Because it wasn't present in the original reaction. And that's because the original reaction I showed you on the leaf was simplified. So you can treat this arrow like an equal sign. If there's 12 waters here and 6 waters here, and let's say you decide to take 6 away here, then you should take 6 away here, and then that drops you to 6, right? And now this looks like the reaction on the leaf, and it's simpler, right? It's got less things to worry about. 6, 6, 6, all over the place, right? It's like a very demonic kind of reaction, but it's also arguably one of the more important reactions on the face of this planet. All right, I hope that made sense. Or you can rewind me, and it'll make sense then, I think. Okay, let's apply the same logic to this equation. This equation is actually the exact opposite of photosynthesis, if you notice, right? Everything has been reversed. This is the reaction you and I do. If you're not photosynthesizing, you're doing this reaction instead. This reaction is called respiration. And respiration is done in the mitochondria by any cell that cannot feed itself. So that includes us, that includes fungus, right? The animal world in general. All right, but let's look at the respiration reaction. Here's sugar. It's got its hydrogens, but if you follow the arrow, sugar now appears as loose CO2 without the hydrogens. So isn't that reversed, like what we saw in the reaction above? Yeah, so we break down sugars, release the energy. And really the reason why we eat a Snickers bar to satisfy us is not for the sugars itself. It's for the electrons contained within the sugars. In other words, metabolism that we all talk about in all circles, at the gym, maybe at physician's office or whatever. Metabolism is all about the movement of electrons. And so chapter 7 and chapter 6, photosynthesis and respiration, are all going to be discussions about how electrons move around. Keep the electrons moving, life goes on. If the electrons stop moving, that's it for life. Life ceases in that form. That's an effective way to die, in other words. So we're going to look at strategies to keep the electrons moving, okay? All right, so if sugar was oxidized, who gained those electrons? Okay, let's look at CO2 and follow the path of CO2. I'm sorry, oxygen. Good grief. See, oxygen here now appears as water with hydrogens that were not present before. They were missing from right there, but now they're present. So that means oxygen gained hydrogens, and to gain hydrogens means to gain electrons, and that means that you were being reduced. All right, so you've learned something really valuable here. What is the major reason why you need oxygen in your life? It's not just to stay alive. Oxygen has a very, very specific role, and its role is to gain electrons. And in gaining electrons, we actually create water. We make water. It's just that we don't make water fast enough. In other words, we dehydrate faster than the amount of water we can create. And that's why you and I still need to hydrate ourselves by drinking. But if we made enough water to hydrate ourselves, that'd be pretty cool. We would be essentially without the need to drink. All right, weird stuff. Email me about that if you want to keep that going. All right, so in summary, in this slide, I really wanted you to understand the terms oxidation and reduction, okay? If you know the definitions, and you can apply them in the remainder of this lecture. And if you start forgetting, remember oil rig to help you out. And also, you cannot have one without the other, and that's what we call it, redox. All right, let's move on. So if we put together those two blank slides where I discuss the two flashcards I want you to create, the light reactions and the Calvin cycle, and then we include the diagram of the chloroplast, and that brings us to this, right? So over here are the light reactions. R, X, and S, that's reactions. And over here is the Calvin cycle. Okay? And again, the light reactions are called the light reactions because those are the ones that need light. The Calvin cycle, notice there's no input of light, but the Calvin cycle still needs things that came out of the light reactions like ATP and NADPH. And do you notice that NADPH here is carrying 2 electrons? Right? And it's basically delivering electrons from the light reactions to the Calvin cycle. And what does NADPH do once it's dropped off its electrons? Well, we can follow its path here. And here it exists, NADP plus, no electrons, no hydrogens. And it goes back to the light reactions to pick up more, and then comes back here to drop them off. So there's a cycle, right, between the light reactions and the Calvin cycle. ATP is doing the same thing. ATP is cycling between high energy ATP and lower energy ADP, right? It drops off energy at the Kelvin cycle, goes back, picks up energy in the light reactions. And here's water providing, do you remember what water provides? Electrons, that's right. And what happens to that water when it's done providing electrons? It leaves as oxygen, 1/2 oxygen. Here it's been super simplified, but it also produces a couple of acid ions. And in the end, what is the goal? That is the goal. The goal is to produce sugar, right? But you remember what is the actual product that comes out of the Calvin cycle? It's not that sugar, it's actually G3P, right? So, okay, so I'm hoping that you're seeing some consistency here. It helps to tie in things. So if this is going too fast and you're losing some things, I think it's a good idea to go back and try not to move forward in this presentation. Or if you do decide to move forward thinking that, maybe something will click in something that I say, try that. But if it only gets worse, then you should definitely go back and then maybe start thinking about writing questions down that I can help you with through email. Okay, I forgot what I was going to do with that slide, so let me go back. Okay, I think I remember what I was going to do, but we don't need that blank slide anymore. Let's talk about light itself, okay? So far we've been calling it sunlight, but we need a better name, especially since we learned about how energy travels in the form we call light, or light energy. This slide shows what we call the electromagnetic spectrum, okay? That's all of this. Okay, I'll write it here. Electromagnetic spectrum. It's basically a long series of waves. It's a spectrum of waves. Some waves are really, really, really long. Look at this one, radio waves. 10 to the third meters is 1,000 meters. So what does that mean? A wave that's 1,000 meters long. It means that energy in the form of radio waves is traveling like this in waves, right? And the distance between one crest here and another crest there, the distance between those is 1,000 meters. Now let's look at the opposite extreme of this one. That's gamma rays. Gamma rays are 10 to the -5 nanometers. That's intensely tiny, right? These are like, I could not draw this and make it fair, so I'm just going to draw something like this. Okay? So the distance between those is like super tiny. Okay, question. If this is the length of a wave, A, and this is the length of a wave, B, which one of these two waves carries more energy? In other words, which carries more energy, radio waves or gamma rays? All right, we should all say gamma rays. The smaller the wavelength, the smaller the distance between one crest and the next crest over, the shorter that distance, the greater the energy associated with that wave. So what do you think about X-rays? X-rays carry a lot of energy. UV rays, they carry a lot of energy. The visible light spectrum, right, which is this. The visible light spectrum is found between UV and infrared, all right? We just blew it up here so that we could see it, all the details of it. We just kind of pulled it out and stretched it out. Let's look at that one now. Here is a wavelength that is 750 nanometers long. And here's another one on the opposite side that's 380 nanometers long. Which one carries more energy? The shorter wavelength, and that would be 380. You know this also. You just didn't necessarily know it in this context, okay? But here's one way you knew this, okay? If I were to ask you to imagine a candle flame and then taking your hand and kind of hovering over the flame with your fingers back and forth, you would feel the warmth, right? But you wouldn't necessarily feel a burn. And what is the color of that flame if not like a yellow, orangey red, correct? That's a candle flame. But now I want you to imagine a blowtorch, right? Sound effect incoming. It's intensely blue. Would any of you dare to take your fingers and go over that blowtorch in the same way as you would a candle flame? Of course not. So you know, right? Maybe you knew by sound, right? And that was a pretty good sound effect of a blowtorch, I think. But you knew from the sound maybe, or maybe from the color of the flame, that that was an intense flame. All right. So the colors, my point is, the colors are now associated with energy. Which colors have more energy? The blues, right? These violet or purple. Which ones have the weakest energy? It's the orange red. And the yellows are somewhere in the middle, right? Okay. So we understand that light energy travels in waves, and that the shorter the wave, the greater the energy. And for that reason, we're now going to replace the word light with photons. What we're saying is that a photon is a wavelength with a specific amount of energy, okay? So if I draw a wavelength, any wavelength, each one of those wavelengths is associated with a specific amount of energy. And for that reason, we no longer call them light waves, we're going to call them photons, okay? So photons are light waves with a specific amount of energy. Okay, we should talk about pigments next, right? So what is a pigment? A pigment is a molecule that absorbs light energy, so it absorbs the visible light spectrum, except for one wavelength. It gives that wavelength away. So as an example, something that appears to be blue is absorbing everything, everything. It's holding it. It's absorbing the red, it's absorbing the orange, it's absorbing the yellow, the green, right? The violet. What is it not absorbing? It's not absorbing blue, it's giving that blue away. It's reflecting it. And for that reason, we get to see it. So if I'm wearing a blue shirt, the reason why you get to see blue is because the pigments embedded in the cotton fibers of my shirt are absorbing every wavelength, the violets, the greens, the yellows, the oranges, the reds, and there's one that it's not absorbing. It's not absorbing the blue. It's giving that away, and that's why your eyes pick up blue. All right, using similar logic, why does a red shirt appear red? Because the red pigments in that shirt are absorbing everything but red. A yellow shirt is absorbing everything but yellow, and so on and so forth. What about white? Well, white is a sort of trick played on our brain when our eyes see all the wavelengths at the same exact time. If you see violet, blue, green, yellow, orange, red at the same exact time, your brain gets tricked into seeing white. You want to test this? Take a white sheet of paper, yeah, cut it into a circle, and then cut it into pie wedges, and then color them. Violet, blue, green, yellow, orange, red. And then, color those in, right? And here, it's going to look like this. Okay, I think you get the idea. Do I have a violet? Maybe. I got a red. I just don't have an orange. But you get the idea. So once you have that on a white piece of paper, attach it to something with a sturdier back, like cardboard. Maybe glue it or tape it. And then take that cardboard with the white paper that's been colored in for the visible light spectrum, and then spin it as fast as you can. And your eyes will see white because you're seeing all the colors coming back at your eyes at the same exact time. Pretty cool. All right, that's white. White is all the colors coming back at your eyes, tricking you into seeing white. What is black? Black is the absence of color. So black really isn't a pigment. It's just nothing coming back to your eyes. And so your eyes interpret that emptiness as black. All right, here's a question for you. Let's see if we understand things. If at the end of, I don't know, a day of work, or at the end of a lecture, maybe in the summertime, you walk out to the parking lot because you want to go grab some lunch with a classmate. Your classmate drives a black car, but you drive a white car. On a hot August summer, which car would you get into? You'd get into the white car, right? Why is that? Because it's cooler. And why is it cooler? Because the sunlight has been blasting it all day with all the colors of the spectrum. And what has the white car done with all of that energy? It's giving it back away, right? That's why you see white. It's not absorbing any of it. It's just bouncing right off the pigments that coat the metal of that car. What about a black car? The black car is going to be intensely hot. And that's because the black car is doing what? To all the colors of the spectrum. It's absorbing it. All of them. Not giving anything back. So all that energy is just staying with the car, making it intensely hotter. All right. So I hope you understand a couple of things here. One, you understand that waves are the way energy travels, and that the shorter the wave, the greater the energy. The longer the wave, the less energy it has. And the second thing you should know is the nature of pigments. How do they interact with light? A pigment of any color is absorbing every color except the one it's showing you, right? So if it's showing you orange, it's absorbing everything but orange, and so on. Cool. All right. What color are plants? What color are the leaves? They're green. And what does that tell you? It tells you that when plants absorb light energy or photons, they're absorbing specifically the photons associated with colors. They're absorbing the visible light spectrum. They absorb everything except what? That's right. They absorb everything but green. And that's why plants appear green. So the pigments that are absorbing all of these colors are found where exactly? Well, what part of this chloroplast is green? Do you remember the name? That's right, it's the thylakoid membrane. The thylakoid membrane has pigments inside of it. And those pigments are absorbing all the colors of the visible light spectrum with the exception of green. So they absorb everything and they transmit green. They give that away. All right, let's move on now. All right, things are going to get pretty detailed. All right, so bear with me. This might be a time to take a break if you need, and then review some things, and then come back because we're going to get super detailed. Let's talk about those specific pigments inside the thylakoid membrane. The pigment of importance is going to be a pigment called chlorophyll. Chlorophyll is what color? Green. And that means it's absorbing everything but green, right? The pigments inside plants perform differently when they're part of the thylakoid membrane versus when we pull them out and they're no longer in the thylakoid membrane. They behave differently. And here's what I mean. If I were to take a bunch of chlorophyll pigments from the thylakoid membrane and collect them all in a flask, such as this one here, and then I shine light on that liquid mixture of pigments, here's what the pigments do with that light. The photons, remember? Because they're traveling at a specific amount of energy, right? This is a wavelength right here. The distance between here and here is a wavelength. So when energy in the form of photons strikes the chlorophyll pigment, one of the electrons in chlorophyll becomes excited. What does that mean? Because we say here the electron is in an excited state. What exactly does that mean? It means that the electron has gone from an inner shell to an outer shell. That's what it means to have become excited. It jumped from being in an inner shell to being now on an outer shell. Maybe this shell or this shell right here. Oh, that's funny. Okay. So what happens when the electron goes from being here to jumping there? Well, this is the part that's different. This is the part that's different between a pigment that's in the thylakoid membrane and a pigment that is outside of the thylakoid membrane. When it's outside the thylakoid membrane, that electron right here cannot stay in this place. It's going to go back to its original position. But going back to its original position means that it has to give away its energy, right? And it gives it away in the form of heat, so it gets warm, and it also gives away another photon. And we call that giving away of the photon, we call it fluorescence. So let's go here. What color is the original pigment? It's green. But what happens to that green pigment when it gives away its energy from the energized or excited electron? It turns red. And in lab, we would have shown you this. We would have taken a bunch of green pigments and we would have shone light onto those pigments. And we would have seen that the pigments go from being green to being red because the electrons are falling back to their original position. So again, I think it's maybe easier to understand here. An electron's in an inner shell, jumps to a second shell or maybe to a third shell, depending on how much energy it absorbed. But when it goes back to its original position, it gives away heat, and it also gives away a photon. And we call that fluorescence. So that's just an interesting thing about these pigments. Okay, so we're going to start adding details here. Here's something we call a photosystem. I'm going to diagram this and explain it again in future slides. But a photosystem, I want you to understand, consists of the following. It's made up of proteins. Some of those proteins are just structural, meant to hold things or to attach to a membrane. And some of those proteins are meant to act like enzymes. Those are the more important ones. All right, so I'm going to put a star on this one. But a photosystem isn't just made out of proteins. It's also made out of pigments. And notice that a lot of the pigments are going to be green. But there's some other pigments in there. Some of them are yellow, orange, blue-green. There's a variety of pigments in there. But the most important pigment has got to be a pigment we call chlorophyll. That's the one we were diagramming in the slide before. All right, so we should understand what a photosystem is. And notice that on this diagram, there are two photosystems. One of them is called photosystem 2. The other one's called photosystem 1. They're also known by another name, P680. You see that here. And this one here is also known as P700. And you see that there. What does that stand for? That stands for the length of the wavelength. So one of these takes in energy that is greater than the other. 680, right? That's smaller than 700. So 680 is a higher energy photon. So the distance here is 680 nanometers. And the distance here is 700 nanometers. All right. Notice here that water is coming in. Here's water. It's going into 680. And what is water providing? It's providing electrons. And what happens to water when it loses 2 electrons? Then water goes on to become oxygen and acid. And what happens to those 2 electrons that it gave to the photosystem? Here they are. They've become excited. And what excited them? Photons from the sun. Notice that photons travel in waves. And when they strike one of these pigments, the wave gets longer. And every time it strikes one of these pigments, it gets longer and longer and longer. It loses energy. So maybe the wave came in at 400 nanometers. That would be like in the blue range. But by the time it's bounced around quite a bit, it's now at 680. And if you go back, if you rewind the presentation and you go to the visible light spectrum, 680 is in the range of orange. That's weaker energy. And that is the specific amount of energy required to excite the electrons in photosystem 2. Those electrons will then be moved across a series of electron transporters that form what we call an electron transport chain. They consist of proteins, proteins with metal ions embedded in them. And metals are very good at transporting electrons. This is why we use copper to help move electrons in a wire to power up devices. Electrons are very good at transporting electrons. So the electron transport chain consists of proteins with metal ions or minerals that help it move electrons from photosystem 2, from here, all the way to photosystem 1 over here. But they go through a chain. So let's practice things that we've learned. If an electron becomes excited, in the previous slide we learned that that electron would just come back to its original position and give off heat and also fluoresce. Oh, I need to tighten this up. I'm starting to fall apart. Right? But it turns out that if the pigment is attached to a membrane, that electron does not come back to its original position. That electron actually jumps from one pigment to something else called a primary electron acceptor. Let me go back to show you there it is, the primary electron acceptor. That literally means the first one to accept an electron. So do you remember now your redox reaction? Who lost the electron? 680. Who gained it? The primary electron acceptor. What is 680? That's chlorophyll. That's the pigment that gives plants their green color. That one loses the electron to the primary electron acceptor. So who was oxidized, who was reduced? Chlorophyll was oxidized. The primary electron acceptor was reduced. Remember your mnemonic, oil rig. Okay, what happens to 680, or chlorophyll, when it loses its electrons? It cannot just keep losing electrons indefinitely. It's got to recover those electrons from somewhere. So who is providing the electrons to replace the ones lost by 680? Water. Again, that's why you water your plant, to replenish the electrons that are being lost by photosystem 2. Okay, so the electrons move and through a process called redox. That's oxidation and reduction happening at the same time. But then the primary electron acceptor loses its electrons to this first electron transporter, and then to this one. And then this guy loses it to that guy, and then that guy loses it to this guy. And then the last electron transporter loses it to P700. How did the electron move around the entire time? Electron. How does the electron move all this time? It's moving through redox. Somebody's being oxidized, somebody's being reduced, right? Primary electron acceptor is now being oxidized by this guy. And then that guy gets oxidized by this guy. And so on and so forth until the electron ends up being here. Okay, let's review material from chapter 2. Why does the electron move from left towards the right? It must be that the right side is more electronegative. Okay, so let's stay on this idea. The role of sunlight, the role of photons, is simple. Excite the electron. Why? To allow redox to take over from there. Boom, 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 the electron moves around. So light is just energizing the electron, making it easier for redox to take place. And redox is going to go from left to right because electronegativity is increasing from left to right. In other words, this guy is more electronegative than that guy. So it's sort of like a vacuum, just sucking the electron from left to right. Okay, so now we're at photosystem 1, and it's carrying 2 electrons that used to be part of photosystem 2. Those 2 electrons are sitting right here on P700. And look, another photon of light comes in really tight and bounces around, gets wider, longer as it moves around, losing some of that energy. So it might have come in at 400 again. But now it's at what? 700. And that's the amount, the specific amount of energy required to excite those 2 electrons again. So those 2 electrons have been excited 2 times. One time in photosystem 2, a second time in photosystem 1. Again, a primary electron acceptor is going to pick up the electrons. And then there's a short electron transport chain here. And eventually, who is the last one to pick up the electrons? Okay, this is a test question. Unit 2, I'm going to ask you, who is the final electron acceptor in the light reactions of photosynthesis? And your answer is going to be what? Look here for your answer. Your answer is going to be, it's NADP+. Now go back and look at your flashcards for the light reactions. And notice that NADP+ becomes what? NADPH with an extra hydrogen, right? So NADP+ becomes what? It becomes reduced. And when you reduce NADP+, the outcome is NADP with an H. Okay, so this was just showing you the flow of electrons. Let's try and put it together in the membrane, right? We'll do simple, and then we'll end it with the more detailed diagram that's in your textbook in chapter 7. Okay, here's the simplified one. If you were wondering, what is NADPH? What is that thing? I'm going to give you the long name of it, right? And you're going to hate it. And we're going to agree that NADPH is just the preferred way to remember it. This is nicotinamide adenine dinucleotide phosphate. You know it as vitamin B3. That's NADPH. And what are the vitamin Bs associated with? Most of you would know, you've seen them. Vitamin Bs are just plastered all over things like Red Bull, Monster, right? Rockstar, all of these. What kind of drinks? Energy drinks, that's right. Vitamin Bs are associated with energy. So we're going to see vitamin Bs all over photosynthesis as well as respiration, and rightfully so. They are electron carriers. That's what the vitamin Bs are doing for us. They're shuttling electrons here and there within cells. Okay, so this is that scheme that we saw in the previous slide. I can go back. Whoa. That same scheme is now here. But we've now added parts of the membrane. We're layering more information here. So here is photosystem 2. And here's photosystem 1. And here's the electron transport chain that connects the two. Oops. And here's one other component that we haven't described. ATP synthase. ATP synthase ends in ACE. What does that tell you? That's right, it's an enzyme. And what does the name suggest it's doing? It's synthesizing ATP, that's right. So here's ADP with a phosphate, and here's now ATP. So ATP synthase is important because it makes ATP. Okay, so let's explore this diagram a little bit more. First, appreciate the location, right? Location, location, location. This bottom part is called the stroma. This is the thylakoid membrane, that's green. And then this is inside the thylakoid compartment. And what we're showing you is, right, if you can see right there, we're showing you basically the bottom of that thylakoid disc, the bottom of that thylakoid membrane. So the stroma, remember, is where you find the liquid that bathes the thylakoid membrane. Okay, so here's what's happening. Light is coming in to photosystem 2, right? These are photons. And they're going to attach themselves, or I'm sorry, they're going to strike the photosynthetic pigments that are part of the photosystem. And electrons will become excited, and eventually those electrons will move from left to right. And who's going to pick up the electrons at the very end? We agreed that was going to be NADP plus, and that's how you make NADPH. Here's those two electrons. Okay, who provides electrons to the light reactions? It's water, right? And when water breaks down, it creates acid, and it creates oxygen. How much oxygen? 1/2, right? There's only one oxygen right there. All right, so light comes in, excites the electrons in photosystem 2. Those electrons that become excited get picked up by a primary electron acceptor. You could see that in the previous slide. Let's go back and look at it. It's right there, right? And then those electrons that were picked up by the primary electron acceptor are then moved through the electron transport chain, and they end up here in photosystem number 1, also known as P700. So remember, this was P680. This one is P700. The P stands for peak. So photosystem 2 likes a peak wavelength of 680. Photosystem 1 likes a peak wavelength of 700. Here's what I want you to get from this particular slide. I want you to understand two things. One, I want you to understand something called chemiosmosis. So in the previous slide, all I wanted you to know was how light energizes electrons so that they can move through redox. And you learned about 3 parts: photosystem 2, electron transport chain, and photosystem 1. You also learned that water provides the electrons that move through the photosystems, through the electron transport chain, that eventually end up in NADP+. So if I should ever ask you in the future, who provides electrons to the light reactions, your answer is water. If I ask you what becomes of water, you will say oxygen and acid is produced. I will also ask you who is the last one to pick up the electrons, and you are going to say that's NADP+, And when I ask you what becomes of NADP+, you're going to say it becomes NADPH. That's all that we learned in the previous slide. In this slide, I want you to understand something called chemiosmosis. You can break the term down to say chemical osmosis. And you know what osmosis is. That's the diffusion of water from high concentration to low through a semi-permeable membrane. So let's see what exactly is chemiosmosis in this diagram. Do you see that when the electron transport chain is transporting electrons using redox? Do you see that there's acid moving from the stroma, which has low amounts of acid, all the way across the thylakoid membrane to the inside of the thylakoid where there is high acid? So if you have a lot of acid, wouldn't we say that the pH here is low? pH is low. Meaning like a pH of like 5 is lower than a pH of 7. And down here, the concentration of acid is low, so this would be a higher pH, right? So here the pH is high. So here's what's happening. As the electrons are moving through the electron transport chain, every time the electrons move from one electron carrier to the next, acid is being moved from the stroma into the thylakoid compartment. Think of it this way. If I were playing hot potato with you, who would hold on to the hot potato longer? Well, it would be whoever has an oven mitt. In this case, here's what I'm saying. If you were playing hot potato with an electron, the electron is hard to hang on to. Once you acquire an electron, you get a negative charge and it makes you very reactive. The way to prevent you from being reactive, if you pick up an electron which has a negative charge, is to pick up something with a positive charge to neutralize the electron. So as the electrons are moving from one electron transporter to the next, every one of them that picks up an electron will also pick up a hydrogen because it has what? A positive charge. And what that does is it neutralizes the electron. So when you pick up an electron, you also pick up a what? A hydrogen. When you lose your electron, you also lose your hydrogen. Except you don't put the hydrogen back where you got it from. You don't put it back in the stroma where you got it from. You put it instead up here, inside the thylakoid. So over time, as long as light is coming in, electrons are moving. And as long as electrons are moving, hydrogens are being moved across the membrane from low concentration to high. Which is weird because we learned that that requires energy. Whenever you move things from low concentration to high, we call that active transport. Which is what's happening here. You're going from low concentration to high. So where's the energy coming from? It's coming from light. That's another reason why light is important. So not only does light excite electrons, but it also allows the movement of acid, H+ ions, from the stroma into the thylakoid compartment right there. And if you do this all day, then what happens to the pH inside the thylakoid? It becomes what? Highly acidic. It's a very low pH. It's actually about the pH of vinegar. So it's not like the pH of your stomach, which is about 2. So why is chemiosmosis important? Why is the movement of acid across the membrane important? That's what chemiosmosis is. It's the movement of acid through the membrane. Why is it important? Because when we concentrate acid, we can then release the acid to go back from high concentration to low. So let's follow its path. See how it's going here? Across and down the ATP synthase. There's the acid now. And that acid is powering up the enzyme ATP synthase so that it can do this reaction. Attach ADP with phosphate and create the second important product of the light reactions. The first one was NADPH. So 1 and 2 products are very important because those 2 products are going to feed the Calvin cycle, and that's a separate lecture. Okay, so I want you to review this diagram. It's the simplified diagram. But I don't want you to stress out in terms of the test because here's what I want you to know. I'm going to give you the diagram that's in your textbook, and I'm going to put that diagram on the exam. So you don't have to memorize things. I don't want you memorizing this diagram. I want you to understand the diagram. And you will know that you understand the diagram if you can successfully write a paragraph or two of the whole process. So here's the diagram I'm going to give you. This diagram is from your textbook. I'm going to put it on your test. So you don't need to memorize this. I don't want you to memorize things and pretend that we learned it. I need you to understand what's happening procedurally in the light reactions. So let's do it one final time. Photons come in. They come in with a high amount of energy. But as they move around from one pigment to another, they lose their energy, the wavelengths get longer, and eventually the wavelength gets to something that can be used by photosystem 2. Do you remember what that wavelength was? It was 680 nanometers. From here, the 680 nanometers energy, that's used to excite an electron. There's the electron being excited, or electrons. It's actually 2 electrons, right? The 2 electrons that came from water. And those electrons are then picked up by a primary electron acceptor. That's right here. And then that primary electron acceptor is going to lose its electrons to the electron transport chain. Notice that the electron transport chain is now drawn a little bit different. It's a little bit more detailed now. It's not just a bunch of even circles. No, it's a little bit more detailed. So this is the electron transport chain. But you notice also that the membrane now that we're drawing is not the bottom of the membrane. We're drawing the top of the membrane. So things are a little bit different. And I want that, in fact, because again, I don't want you memorizing stuff. So I want you to compare this diagram to that diagram. This is the bottom of the membrane that we drew here. But on this diagram, we're focusing on the top, not the bottom. And that makes a subtle difference to some students, right? We'll see if you can make sense of it yourself. What happens when electrons go from photosystem 2 to photosystem 1? They have to travel through the electron transport chain. And in doing so, we move ions from the stroma, which is here, and here. We move acid from the stroma to the inside of the thylakoid compartment. And notice that there's less acid here, and there's just a ton of acid in here. All of that is because of that transporting of acid into the thylakoid disc. What happens to all that acid eventually? It goes down the ATP synthase. So here's a better, more detailed illustration of the ATP synthase. Acid goes from high concentration to low, and that energy is used to create ATP. Where will that ATP eventually go? To the Calvin cycle. That's lecture 2. And that's going to be a lot kinder, a lot easier, less detail. Who is the final electron acceptor? NADP+. What happens to it? It becomes NADPH. And where does that go? Also to the Calvin cycle. Okay. That was quite a bit to finish off with. But the beginning was probably not that difficult, okay? So I expect to hear some questions from you. Here's one thing that I'd like to initiate in a discussion. You can just chime in that discussion and say, oh yeah, I got that, or no, I was confused by that, but I understand it now, or no, I'm still confused by it. Let's generate a discussion. I want somebody to initiate it by telling me the difference between antenna pigments, antenna as in literally an antenna. So I want you to tell me the difference between antenna pigments and chlorophyll. Let's generate a discussion, all right? I expect somebody to start it off, and then I want all of you to, at some point, chime in. You don't have to, say something that is like earth-shattering, right? I just want to hear from you. I want you to check in through a discussion, right? So I'll go into Canvas and I'll try and set something up. All right, everyone. Hope you're following along, and if not, then I expect to hear from you either through email or through Canvas. Okay, I think that's it for the light reactions. Take care.